Welcome everyone. My name is Craig Garner and this is my second presentation on the topic of anthropogenic causes of increased corticosterone levels. The scientific paper that we will be reviewing is titled, "Does Urbanization Cause Stress in Wild Birds During Development? Insights from Feather Corticosterone Levels in Juvenile House Sparrows." This paper was published in October of 2018 in the scientific journal called Wiley in the Ecology and Evolution open source section. Before we start discussing this scientific paper, I thought I would start with a little bit of background information on house sparrows for those of you that aren't familiar with this species. However, I would imagine that if you've lived in urban areas, you probably are familiar with this species or you have seen this species, whether or not you recognize it or not. On the right hand side of this slide, I have two pictures. The top picture is the slide of a female house sparrow, and the picture in the bottom right is represents a male species of house sparrow. As the name indicates, its common name house sparrow is indicative of habitat that it utilizes worldwide. It's a very common visitor or user of habitat in urban and residential areas. If you've had a bird feeder in your backyard, you've probably seen this species quite commonly, and it's probably the most popular bird at your bird feeder. In the wild, these birds generally live to about 3 years old. However, the oldest species was recorded in Texas in 2004, and it was estimated to be approximately 15 years and 9 months old. They have a near worldwide distribution, and the areas that they were excluded from are primarily extreme weather areas. These are areas in the far north that are very cold, or areas that are very hot and dry, like deserts. This is a non-native species in North America. It was first introduced through Brooklyn, New York in 1851. And currently there's a population size of approximately 540 million breeding pairs worldwide. And it is classified as an urban exploiter. As I mentioned, it So why is this study important? House sparrow populations have been and are declining worldwide. The rate of decline depends on what part of the world you are looking at, but the common consensus is there is a decline in population numbers. There could be a number of reasons for this decline, including habitat loss, climate change, introduction of invasive species that creates a shift in food resources, and urban-induced stress constraints. Previous studies have found that urbanization can cause behavioral, physiological, and morphological changes. This includes increased levels of corticosterone levels and associated constraints. As we've learned in our previous lectures, that increased corticosterone levels induce short-term behavioral responses, and long-term exposure to these responses could create a reduced reproductive behavior, increase nutritional stress, it could reduce body mass and size, and reduce feather quality. To better understand the role of corticosterone levels in house sparrows and what role this could potentially play in population declines, the goal of this study was to understand the connection between urban stressors and corticosterone levels in juvenile sparrows. The second goal was to understand the effects of increased levels of corticosterones on body size, body mass, and body condition, and to better understand the link between corticosterone levels in feathers versus plasma. The study area for this project included 11 sites throughout the country of France. In the upper right-hand corner of this slide is a figure 1 from the paper that represents the location of the study sites. And as you can see, they were sparsely spread out throughout the country along the east and west side of France. At each site, the habitat was classified as sparsely populated or highly urbanized. The level of urbanization was quantified using the techniques developed by Leiker et al. In 2008. The process involved using aerial photographs and dividing each site into 100 evenly spaced cells. Within each site, 5 habitat categories, including building density, number of cells with high density buildings, vegetation density, the number of cells containing high density of vegetation, and the number of cells containing roads were classified. The principal component analysis was used to assign an urbanization score for each capture site. The principal component analysis is a statistical procedure that converts a set of observations that are possibly correlated into a set of values of linearly uncorrelated values called principal components. These principal component scores were used to assign the urbanization score for each of the capture sites. And a total of 111 juvenile house sparrows were captured using mist nets at the capture sites. This project collected data on feathers, morphological measurements, and blood. Feathers were collected from all 11 capture sites. Directices, these are feathers in the tail, and the 2 feathers closest to the middle of the tail were collected. The image in the upper right-hand corner of this slide shows an image of a bird with the different feathers and the names for those feathers. And if you'll notice, the tail feathers are called rectices. And if you look very close, the 2 middle feathers are each labeled R1 on this image. And I believe these are the 2 feathers that were collected in this study. After collection, the feathers were stored in dry paper envelopes for further analysis. Morphological measurements included weighing and measuring all of the collected feathers, body mass, wing length, tarsus length, and bill length were measured for all juvenile sparrows at 4 of the 11 capture sites. Fat and muscle scores were collected per methods of 3 previous studies. Blood samples were obtained from all juvenile sparrows on 4 of the 11 capture sites. Samples were obtained within the first 3 minutes. The very first samples were obtained within the first 3 minutes of the capture. After the first sample, the specimens were stored in cloth bags, and a second blood sample was taken after 30 minutes of capture. The sample taken within the first 3 minutes of capture was used as a baseline sample, and the 30-minute sample was used to measure corticosterone level increases. A 25-gauge needle was used to draw the blood, and the blood was stored in microcapillary tubes containing heparin, and this heparin was added to prevent coagulation of the blood. And the blood was taken from the Aller vein. I'm not familiar with this vein. I Googled this and could not come up with any information on the Aller vein, so I'm not sure if this is a mistranslation in the report or maybe a misspelled word, but the ulnar vein is located in the wings of birds, and my assumption is this is where the blood sample was taken, from the ulnar vein in the wing. But if anybody knows, clarification on that would be appreciated. Once the blood was taken, the samples were centrifuged at 4,500 RPMs for 7 minutes to separate the plasma and the red blood cells. And the plasma was used to measure the corticosterone levels, and the red blood cells were used for molecular sexing of the juvenile birds. Juvenile body conditions were analyzed using the scaled mass index method. These methods were described by Pegg and Green in 2009 and 2010. Tarsus length was found to have the highest correlation with body mass, so it was used in the SMI formula that you see on the screen, where MI represents the body mass, LI represents the tarsus length, LO is the arithmetic mean value of tarsus length for the entire population, and B is a slope estimate of a standardized major axis regression of log transformed body mass and log transformed tarsus length. The sex of juvenile house sparrows were identified using the molecular sexing technique described by Fridolfsson and Elgren in 1999. The male DNA sequence consisted of the CHDIZ sequence, and the female DNA sequences consisted of either CHDIZ and or CHDIW sequences. The corticosterone analysis consisted of measuring corticosterone levels using the radioimmunoassay techniques. This consisted of using 10 milliliters of methanol and adding this to each feather to extract corticosterone from the entire feather. The feathers were then stored at 50 degrees Celsius overnight in a shaking water bath. The methanol was separated from the feathers and the feathers were washed. The feathers were then added to the original methanol extract and were analyzed for corticosterone level increases. The study found that there was a positive relationship between the urbanization score and the level of corticosterone in the feathers of juvenile house sparrows. It found that for most sparrows living in urban sites, the level of corticosterone was approximately 20% higher than the specimens living in rural sites. In the upper right-hand section of this slide is Figure 2, and this figure represents this relationship. You can clearly see that as the urbanization score increases from a -2 value through a +4 value, you see a positive relationship and an increase in feather corticosterone as the urbanization score increases. The study also found that there was no relationship between the standard mass index, the tarsus length and body mass with corticosterone levels in the feathers, the sex of the specimen, or the corticosterone and the specimen sex interaction. The study found that neither the baseline or the stress-induced corticosterone levels were related to the feather corticosterone levels, the sex, or the corticosterone and sex interaction. However, the study found that the corticosterone in the plasma in juveniles was positively related to the feather corticosterone levels, and this did not differ between males and female specimens. This study demonstrated that as the level of urbanization increased, so did the corticosterone levels in juvenile house sparrows that occupied these urban areas. Since juvenile feathers are in the early stages of development, the study results indicate that increases in corticosterone levels and the associated constraints likely occurred while the house sparrows were in early stages of development prior to fledging. These results contradict previous studies. However, those studies use specimens post the juvenile feather molt. Therefore, the studies are not 100% comparable. The study results supports the hypothesis that urban juvenile house sparrows are exposed to stress-induced constraints during development, and this may lead to reduced survival rates. However, this study cannot confirm that there is a reduced survival for urban dwelling house sparrows. However, numerous studies have documented a negative impact of urbanization on house sparrow populations. In addition, numerous studies have found evidence of population declines, reduced reproductive recess, reduced body size, and feather quality due to stress-induced constraints from urbanization. However, this study did not find a positive relationship between corticosterone levels and body size. As we learned in previous lectures and from this study, moderate levels of corticosterone for short-term exposure can be beneficial. The results of this study could be interpreted to suggest that urbanization may only be a constraint at certain levels of urbanization. However, determining at what level of urbanization does it become a negative constraint is beyond the scope of this project. The authors suggest that additional research and studies that examine this relationship would be beneficial, and I agree. Understanding this relationship in more detail and determining at what level does urbanization become a negative constraint would provide critically important information for conserving this species. I hope this presentation and my previous presentation was enjoyable and provided enough information to help make the articles easier to understand. I look forward to the upcoming student presentation, and for all the students working on presentations, good